everybody's revived with a little caffeine. All right, so this next set of slides is going to be showing you a really cool tool that we've developed over time. We've tweaked it over time. And this tool is the Immunome browser. This is a unique tool. This is not something that you find, well, it is, but we'll talk about that in a minute. It's integrated, it's a tool that's integrated into the query interface. So you run a query and then it'll map the results to your query. So what is it? The Immunome browser maps the immunome, and what we mean by that is that it allows you to visualize the totality of immune responses from that host, on that protein, T, B, so forth, and I'll show you that. So it maps the responses to an individual protein, or in the case of a virus, a polyprotein. So what is it for? So very often we get asked the question, what's the immunodominant epitope? What are the best epitopes, right? And the way to get at this is not trivial, because it depends on the host, right? What's immunodominant in mice is not immunodominant in humans, and even among human populations, there's different ranges of dominance. It also allows you to look at what areas of an antigen are well covered and which are not, or which are immunogenic and which are not, depending on the responses. Where is it located? It's located on the Antigen Tab in the results page. And how does it work? So what it does is, for a given query that you run, it plots the response frequency score for the epitope along the entire length of the antigen. And it does this per residue, and we use reference proteomes or reference proteins for that, and these are from NCBI. So you're not just mapping where the response is, you're mapping the relative prominence of that response. So how many times that residue was recognized by different hosts. So it's got meaning from an immunological standpoint. So I'm going to do a demonstration. The demonstration we use is HCV. HCV is a virus that is extremely well covered. All right. So the genomic polyprotein is 3,000 amino acids, but if you do a query, you get a result of over 4,000 epitopes. Does that mean this is all crazy nonsense? No, it just shows the abundance. So there's iterative coverage. Many labs doing many different experiments in different hosts and different scenarios. And so you get an abundance of epitopes, so you get a lot of coverage. This allows you to look at this coverage, and if there's a tremendous amount of data, as is in the case of HCV, you can look at the choice trees for the forest, so to speak, because you can give input criteria that's very specific to your needs. All right? So you use the selective criteria. You can either look very broadly or very specifically. And in this example, I'm going to look very broadly. So we're going to go to the home page here. And in this case, I went to the antigen, and I typed in under organism HCV, and I got my autocomplete coming up. And I selected HCV, and I hit the search button. Then on the results page here, you see under the tab, as I mentioned, it's under the Antigen tab because it maps it to the reference proteome. You see the icon here, and you saw this, Bjorn showed this to you as well. If you click here, then it's going to map this data. So you can see there's 4,600 some epitopes. There's tens of thousands of assays coming from nearly 600 pages. So this is a very well-covered virus. So when we do that, you're going to see two things, and Bjorn showed you a snapshot of this. You're going to see two graphs. The top graph is the response frequency graph. This is the response frequency on the y-axis, and the epitope residues along the x-axis, so y and x-axis here, and this will tell you what it's being mapped to here. So you can see I overlaid the schematic of the proteome from HCV to show you where the proteins lie, so you get an idea. This does not come with the output, but you get an idea. Oh, I'm dying here. Oh well, let me use this. All right, so the top graph shows the response frequency score. So you can see this is the upper and lower bounds of the response frequency score over the length. So you can see, for example, core protein. And remember, this is all responses. So this is all responses for so all hosts. All right. Then the second graph you'll see on that page when it pops up is you'll see the assay count page. This allows you to look at both positive and negative. So on the upper graph, you may see a blank spot. You don't know, is it blank because it's a non-immunogenic region, or it just hasn't been well covered? This graph allows you to answer that question. So you get the relative counts for assays. Then if you scroll down, you get the data table. So the table, this is representing all the data that you see in the above graphs. So I'll just walk you through this briefly. This is the epitope ID. As Nina mentioned, every epitope receives an ID. There is the epitope here, and here it is listed by position. Then it shows you the identity, the reference proteome that it was mapped to. It gives you the actual raw data for the number of respondents and the number tested, positive, negative assays, and then here the response frequency score. And then this is exportable in Excel. And then once you export it in Excel, you can then, for example, sort on high to low response frequency scores. And I'll demonstrate that a little bit later. All right, and I'm going to spend a few minutes here. We now have a standalone version of this. This has been just recently published. This is an Immunome browser tool that let you to look at your own data. So this is user-supplied data. So you can supply your own epitopes and response frequency scores, and then give it the input of which protein you'd like it to map to. I've added here the PubMed ID of the paper that was recently published, so you can read that paper. And then this tool is located on the drop-down menu under epitope analysis tools, or it's located at this specific address. All right, and Sandeep is the person to talk to about this if you want to touch base with him during breaks or lunch. So I want to spend a minute, you've heard a little bit about this, I want to spend a minute talking about two important subjects. Cancer, as Bjorn pointed out, programmatically it's not in scope for the IEDB, but the IEDB does contain cancer data that's related to pathogens that cause cancer, and also mass spec dilution data. This is ligand dilution data. So just a brief how you would find these if you were interested. You can, on the main page, you can search by the antigen. For example, you type in the antigen, the cancer antigen that you're interested in. Or you can search by disease. You can look up different types of cancer. Or on the specialized search page that Nima showed you under epitope details on the drop-down menu, you can look at Epitope Hub, and that'll give you a list of the cancer papers we have today. So we're not a repository of cancer epitopes. We want to make that clear. However, we do have some cancer data, and fingers crossed, going forward, that might become a reality. All right, another topic that's near and dear to my heart is the idea of using the database for vaccine development. Right, many of us are interested in that, and so in many of the meta-analysis that I complete, one of the major tasks in that for me is finding a subset of epitopes that are the cream of the crop, and that is ones that would be useful for vaccine design. So what I do is I select specifically on human data, and then using this would be a specialized search category of query. I want to look at all of the epitopes that have been recognized in the course of natural infection. So human subjects with dengue fever recognize this epitope, or hosts vaccinated with this vaccine who recognize an epitope. So that kind of data. All right. Other data is data that reflects immune correlates of protection, so in vitro correlates of protection, B cell responses like neutralization, CTL response, gamma interferon data, and again, that would be an assay-specific search. Also looking at assays that are demonstrating protection, so epitopes that are associated with protection, so in vitro correlates of protection and in vivo correlates of protection. All right, and then finally, I also use the Immunome browser. As you can see, you can look at, for example, the core protein. If you remember the core protein, the N-terminus of that genomic polyprotein was a heavy hitter for both B and T, also envelope. So you can get a sense of that from the Immunome browser when you specify just mapping human data. All right, so now we're going to go into some example queries, and we're going to do this live. So I'm going to show you the first query that we're going to run. And this is, I'm just going to do a linear search using a peptide derived from the nucleoprotein of flu A. Right, and the elements that are going to be highlighted is doing a sequence search, running a homology search from that initial sequence input, the filter feature, drilling down into the specific data. We use the term drill down again, that's jargon, but getting you familiar with the results tables as a summary, but also getting down to the details. All right, so from here, I'm going to go to the database. So you can follow along, or you can simply just enjoy. All right, so I'm going to type that in, and I'm going to hit search. So I'm going to try to walk slowly through this. Let me know if I'm going too fast. So you can see the results for that query are here on that Epitopes tab. There's a total of 5 sequences that match that linear sequence, exact match. I'm going to walk through this. There's the Antigen Tab showing the nuclear protein from influenza, and it shows you the number. So again, you've got these summaries here, the number of epitopes, the number of assays, the number of references. You've got the assay summary page, which is a little bit more busy. So again, we've got an internal assay ID here. This is the reference that it came from. So this is what this row represents, one assay. This is the epitope and the protein in the organism, summarized right there. The host, the immunization process, whether it's an administration or primary induction in vitro. The assay antigen, in this case, here it was the epitope, here it was a related peptide, so other. The restriction that was defined in the assay, and then the assay itself, the type, and then a qualitative, positive or negative, all right? So things you can do is you can sort here. So you can sort on these, and these are sortable, all right? So you can here, do that. Also, you'll know there's pages, so you can go page here. These are just elements I want to touch on. You can change how many you see per page. You can export this. This is the export, all right? And I'll demonstrate an export in the subsequent examples. I have receptor, so this is the receptor data here. And again, that's exportable. And then the references here, 145 references here. And you can see you've got the authors, the title, so just a summary, the abstract, listed by most recent. Here, this is an internal, the reference ID, as Nina indicated. The other thing that's neat, remember I told you how you can use this as a quick way to get what's known. You can quickly go to PubMed, click there, and you got your paper PDF. So you can very quickly get the information by linking to these other databases, all right? So just from there you can get that. All right, so let me walk back here. All right, so you notice that there's these other epitopes that came up. What do they mean? Well, these are the same sequence, so it's the exact match sequence. However, these have post-translational modifications. And if the authors report those, we can capture those. So in this case, this is a glycosylation at M6, a glycosylation at E4, and a main chain modification, which is probably why these are showing up as analogs, because they're probably not a naturally occurring peptide, so they don't map to a naturally occurring protein. But that you see the summary, all right? So, of course, the vast majority of the data is coming from this naturally occurring NP peptide. All right, so now the next feature I want to show you is the filter function, all right? So if we were, since you get a tremendous amount of data, but you're just interested in this one and you don't care about these other, you can simply click here on the funnel, and then you'll see the tabs will reset for just the data related to that particular epitope, right? All right, so you've done that, you've looked at it, so now you click that off, refund the search, and then we're back here, all right? And then this is going to show you how convenient it is to have the recapitulation of these search interfaces here. Now I'm going to say, well, I'm going to look at peptides that are homologous to this. So I'm going to say, give me all too much copy. Give me all data at 70%. We run that search. And now you see there's many more peptides at a 70% homology to that initial input, all right? So you can look down here, many of these will be natural variants of the flu, NP. NP does vary, not as much as HA, but it does. And then say you want to look at one of these look like they're probably analogs of that protein. So let's pick one here, let's look at just the data for that. So I'm going to fixate just on that data. All right, so now we have that. Now you can peruse this data, so you can say, okay, well, there's 2 assays. The assays tab is subdivided, I did mention this before, into T-cell, B-cell, and ligand. Again, that would be binding assay and elution assay under the high node. Let's say you're interested in this particular assay and you want to see the details of this. And this gets down to the details page, right? So I'm going to walk you slowly through the details. So all these detail pages are organized the same way. The top bit is describing the paper. And again, you can link to NCBI here. So this is the authors, the abstract, the PubMed ID. The next section is devoted to the epitope, so it gives you all the information related to the epitope. The next section is related to the host. In this case, it was black snake mice. They were administered the influenza A virus, so the organism. There was an in vitro restimulation step using the peptide, in this case, using splenocytes. This is annotation related to the immunization process. This is information about the assay itself. So this was a Kerman release assay, finding cytotoxicity. It was positive. The effector cells were splenocytes. And we can describe whether they were stimulated, whether they were clones, whether it was direct ex vivo, and all of that is searchable too. So if you're just interested in things that were defined directly ex vivo, you can do that as well using specialized search. So antigen presenting cells, in this case it was a cell line, EL4. The epitope on the antigen in the assay was the epitope itself, so the peptide. There's details here, more annotation that is specific to the assay, descriptions of that. And then finally, what figure in the paper. So if you grab the paper by clicking on the link, you now have the paper, so you can go straight to the figure, so it's very handy. All right, so that is that example. So that shows you a couple of features, how to do a sequence search, homology search, filter down, and grab details for that. All right, so I think now Nima's gonna do his.
Can I just ask a quick question?
Yeah.
Sorry, I
I'll
know you
let
posted
Nima
out. On your, so when you did your initial search and then you have the variants with whatever post-translational modification, are those data actually included in your top level, the one with the highest number of hits, or are they not?
No, that row with all the data, so if I hit the funnel for, say, the main chain modification, the first one, all of the data would be just for that.
So no post-translational modifications in the...
Let me say that again.
Yeah.
When you did the initial search and you have the 5 epitopes listed on the tab, if you were to look at the assay data, they would be there as well.
Okay.
So it's data for all of those.
Okay.
So if you want to just look at the one without...
If we have time, can we just do that?
Pardon?
If we have time, can you just show her? I think it's always easiest to...
No, that's a good question, because those are details of the, that get to the complexity. Where am I? Firefox. Thank you. All right. All right, yes, so what she's asking is, when you run this initial query, so the data that is here is for all 5, yes. So to just get the data for that, you would funnel, yes, that's a very good question, thank you for asking, yeah. All right, so I'll hit this for Nina.
I have a question about the homologs.
Yes.
So
The analog, the non-natural, yeah.
well, you put up the variants of the linear sequence. Those are polymorphic sequences that were similar to the linear sequence that you put in.
Yeah.
Is that determined from the literature as well? Those are related.
Let's see if I understand. So the initial one I put in was a wild type, naturally occurring peptide.
Yes.
And then I ran a homology search for anything that was 70% sequence, homologous at the 70% level sequence of that original sequence.
Yes.
So that'll pull up anything, including analogs that are non-natural.
In the literature, is that also literature-based? Is it empirical?
Yes, so
It's
that,
been found
what
in the
comes up, what comes up are epitopes that have been tested, yes.
Yeah, that's not just taking the sequence and doing a 70% homology.
No, thank you, no, yes, that is of the data, what in the database matches that, yes.
So it is calculated on the fly.
It's
not-
calculated
I
on the
think
fly,
what he's
but
asking is, are they defined in the literature as analogs? And not necessarily, right? So it might be somebody else just studied something else that has 70% identity. So it's not,
yeah.
the fact that it's an analog is not coming from the literature. We're doing a
Whose
separate
algorithm do you use for that, for the 70% from all?
That I'm not sure. Is it the calculation is?
Probably just a Hamming distance, isn't it?
Yes.
Yeah. Yeah. But the hits that come back have been tested in advance. Yeah. Good questions. All right, Nima?
Yeah.
Okay, so I'd like to walk you through an example of a B-cell detail search, since earlier the overview was not so specific and just more acquainting you with the fields. There were a couple of attendees who indicated interest in Ebola. One particular person who indicated that they work in the field of Ebola, so I chose Ebola as an example of using the detail search, and specifically the query that I formulated here is find Ebola virus epitopes recognized by neutralizing human monoclonal antibodies. So all of the important parameters of this query are, of course, defined in the literature. So that we captured it as a neutralizing epitope because the antibody was shown to bind to that peptide and be neutralizing. And so briefly, you should have access to these screenshots as well, so I'll just briefly walk you through the steps to familiarize you, and then I'll also do it live just to give the full effect. So we're going to use the B-cell detail search from the specialized search menu, which will bring us to this interface, and you can always confirm which one you're on by looking at the heading of the fields, because they do all look pretty similar. And so in this case, we will expand the epitope related fields and choose Ebola virus. So this Ebola virus into tree with that ID is a top level organism, as you'll see when I can open the tree to show you that. But in this case, we're doing a very broad query for all Ebola virus epitopes. Then on the host side, since we're interested in human antibodies, we can define the host as human, and then go on to indicate positive, because there are certain, under the qualitative measurement, we will indicate positive because there are certain situations where we might have monoclonal data that is negative. So as a general theme, I don't think we talked about, anyone mentioned this yet, but as a general theme, we don't always capture negative monoclonal data, but there are some cases where we will, at our sort of discretion and based on certain guidelines, make exceptions to that rule. Specifically, let's suppose a particular monoclonal antibody was shown to, let's say, neutralize particular strains of a virus and then not neutralize a certain third or fourth strain. That's interesting to contrast that and potentially make that available for a query because it's maybe not expected, or the absence of that neutralization might be of interest to the user as well. So in this case, since I'm going for true neutralizing antibodies, by clicking positive, you'll ensure that you're not going to get any negative results. And then I will define the assay as we have an assay specifically called neutralization, so we'll delimit the responses by only those assays characterized as such. And then finally, in the assay side, the assay cluster of fields, you see we have a field called antibody purification status, and this drop-down allows one to delimit, again with multi-checkbox capabilities, what level of purification you're interested in. So I chose monoclonal here, but you can also choose some of the other monoclonal choices if it's of interest to you, for example, display library. So by doing that, you have enough parameters to run the query, and I think here I'll just switch to the live. What you see there are screenshots of the actual results, but we can run this in real time and then look at those results tab by tab. Okay, so here under the Organism Finder, to define the source organism of the epitopes of interest, This is where I chose Ebola virus. So as you type, one benefit here is that it's, you know, like the kind of Google style suggestions come up. So that's nice. You could alternatively also use the Finder that Carrie and others this morning have shown you and browse the trees to see the kind of hierarchical relationship between all the different levels of Ebola. But in this case, since I kind of have in mind the target, I'll just run with Ebola virus. And then as I showed you earlier, we're gonna delimit this to Homo sapiens. So again, as you start to type, your hits come back and you can delimit it when you see the choice you like. Somebody asked earlier about multiple selections in these types of boxes. So for example, if I'm also interested in mouse monoclonal antibodies, you could choose the mouse here, and then as you'll see, your selections will basically be stacked. And in theory, you could keep doing that as long as you wanted, and then go back and remove them one by one as you desire as well. So here I'll just keep it at human. And then we're gonna go down to the assay field. And as I mentioned before, sticking to positive is the goal here. In the assay finder, we can again take advantage of the autocomplete. Now that you're aware that there is a neutralization assay, or even if you weren't aware, you could always kind of go on sort of a fishing expedition and type things in and just see what you get, right? But of course, the best way to acquaint yourself with the assays we have are to peruse the tree. And then for future use, the autocomplete, as we call it, is a good option. And then under the assay to antibody fields just below here, I can expand that and you can see various parameters of interest come up in terms of the purification status, isotypes, different chains, etc. So we're going to use monoclonal for this particular query. And I believe that was it, right? So by executing the search, we get a total of 15 epitopes. And going over to the Antigens tab, you can see again, because we used the higher level Ebola, the actual antigens come from two different, what would you call these? These are not strains, are these species? So forgive me, I don't know the complete taxonomy of Ebola virus, but we did take the high level, so these would be found under the tree of Ebola virus. And as one might expect, since we're looking for neutralizing antibodies, all of the hits come from antigens that are like, you know, glycoprotein surface antigens. But again, you could go to the assays tab and look at the particulars of each assay. So I'll just choose the first one, go down to what we call the assay details. So anytime you click on one of those records, you always kind of have a similar layout in terms of the theme of how the fields are laid out. So you'll begin by seeing the reference, and here you're seeing much more detail than you see in the tabular summary form in the previous slide or previous page. So you have a complete abstract, everything essentially that PubMed lists as far as affiliations and title, etc. The journal, but down at the epitope box is where the curated data really begins. So in this particular case, we have actually a discontinuous, again, I think if we look back, we probably see that many, if not all of these were discontinuous epitopes, as they often are in neutralizing monoclonal cases. But in this case, you see one residue from the Zaire glycoprotein. And if we go down to where the assay information begins, we can see again in the immunization cluster fields, these will be various lengths depending on how much data was actually captured and available. So in this case, we have Homo sapiens as the host. For example, if the manuscript would have indicated that these donors, for example, were from a particular geographic region, we would have that as well in this area. Oh, actually we do. I'm sorry, I apologize. It's down below so you can see West Africa. These were, and these may not be so specific all the time. They may be broad geographic regions, but we could even go down to the level of a city. In the process types, which you heard about, so these, in this particular assay, the host, the human, was an Ebola hemorrhagic fever patient who had recovered. So the disease stage, we call it, is post, showing that their blood was most likely taken after recovery. And so because they're naturally infected, we expect the immunogen to be source organism. And this is also important because when we use source organism, what it implies is that the paper explicitly or implicitly by the geographic region said that they were exposed to Zaire Ebola virus. So in the case where we don't know that, we might make it more generic and say this person was exposed to Ebola virus, but we may not know the particular species, so we would not assume that it was Zaire. And that is, these are examples of the comments fields where you get more enumerated details about, if available, about when the blood was collected. For example, in this case, it was during the 2014 Zaire Ebola virus outbreak. And of course, because we specified monoclonal antibodies, we can confirm here that this is in fact an IgG monoclonal that in this case was shown to neutralize not the organism that's the source of the epitope, but a taxonomic sibling of that organism. So in the case of the epitope, the source was Zaire, correct?
Mm-hmm.
And here in the assay, they show that it neutralizes resting Ebola virus. So taxonomically, those are of course related. They're at the sibling level, and this is one of the other powerful things that briefly came up before when somebody asked about cross-reactivity. The database can be used to look for. In fact, you could also go back and see whether this particular monoclonal antibody did in fact neutralize or react with other species of Zaire or other organisms, like for example, if you pull the camera back and you go into the whole flavivirus, as the example came up, you might not even be necessarily at the species level of taxonomic relationships. So anyways, we can go back and see that there are also assay comments in this particular case. We do at times capture commentary about the assay to kind of, again, add more detail, maybe certain things that there isn't necessarily a specific field devoted to, but we find of interest or potential value, we'll capture in there. And then sometimes it's just to clarify what you see in the fields. So for example, in this case, you see this comment says that epitope-specific monoclonal antibodies were shown to neutralize VSV encoding GP. So it tells us that in this case, the as we call it, the antigen-containing object, you had a recombinant organism that was actually expressing glycoprotein from restin. So it cross-reacted across species to the other glycoprotein, and that was expressed on this recombinant vesicular stomatitis virus. So this clarifies what you see kind of on the antigen side. And of course, the location, if you ever were to go back and want to see the data or read the manuscript and see where this data was characterized, everywhere that it is cited here in the location fields of the data in the reference, in the manuscript, that means.
Is there a way to actually query for, say, antibodies that recognize taxonomic siblings?
Yes, there is. So if I go back to my query, I can, using the antigen fields, here, see where I am here,
Yep.
the antigen field, and I can expand that. And if you recall earlier, we had talked about the relationships. So if I go to epitope relation, there is epitope relation tax sibling. Now, if you use this, taxonomic sibling just means that whatever molecular entity or biological entity that the cross-reactivity to, and what's to, was from a taxonomic sibling of the source of the epitope. As you saw, it's not necessarily going to be an organism. It could have also been a protein that was from an organism that was a tax sibling. And, we see that very often. So, but yes, you can certainly do that. Here you have another choice might be that if your epitope is at the higher level of broad, let's say Ebola virus, you could use tax child, and this implies that you want to see essentially every taxonomic organism that's a child of Ebola virus. So that might be, in some ways, even a more powerful query if you're trying to go from broad to specific. Okay, so going back to any other questions? All right, so then if you want to go back to my slides. Go ahead. You can tell I'm a novice Mac user. How do I minimize this without closing it?
Just go down to the bottom and just follow the slides. That'll be the fastest.
Yeah, it's not really, I think I need to, here we go, thank you. It's finicky, it doesn't always come up.
Yeah, the mouse is a little.
All right, so that was the end of my query. I think we go back to you now, query, right?
Yeah.
Yes. Would you like your slides or the?
We can leave it there for a second.
Yeah.
Thank you. Okay, so more live demo. So this is designed to talk about a virus, to demonstrate many features of the database live, using a virus that many of you are very familiar with. So how would you best use the results? How would you navigate? How would you download results, and so on and so forth? So I'm going to walk you through this. So we'll go to the main page. Using the mouse. All right, so here I am on the home page. So, because I promised to do so, first I'm going to do this. So I'm going to look at flavivirus. All right, so autocomplete gives me flavivirus. So if I wanted to look at not just dengue, but what else besides dengue, I'll run a query on that. So you can see there's a tremendous amount of data for flavivirus. So if you're interested in individual organisms, you can look at, you can peruse that here. So hopefully, the fella, there you are, there you are. So this is where you can get at that. So what you can do is you can use the funnel to specify that you're interested in one versus the other, and so you can look at the different, and plus the data is summarized here. So again, these sort by number of epitope or number, the default is sorting by the number of references. So here you can see for dengue, there's a tremendous amount of data, but there's also data for yellow fever, West Nile, Zika, JEB, tick-borne encephalitis, down the line. So that gives you that. So does that answer your question? Hopefully, okay, great. All right. So I'm going to go back. I'm going to reset. All right. So I'm doing this from the home page because I want to show you that you can do a lot of this from the home page. But so I'm going to do a search on dengue. All right. So just all dengue virus. So I'm going to run that search. Can everybody hear me? Am I take for granted that people can hear me? All right, so as you can see, you see that same result as when I just filtered on dengue on the previous query for flavivirus. So there's nearly 7,000 epitopes. So you can see there's a tremendous amount of data. All right, so again, above you see the current input parameters here. So that's kept track. I want to remind you that that's there. The Antigens tab is here. So you can see there's the Immunome browser. There's the Assays tab here. Receptor data and references. All right, and again, you can click on that, grab the paper, that particular paper that's just been published by that group, and click through. Now, the first thing I want to show you, so it was a very simple query, it was just host. First thing I want to show you is how would you download this data? So for the vast majority of the analysis that I do, I always download the Assay tab. And the real reason I do that is everything is on the Assay tab. So the information about the epitope, the information about the antigen, the information about the reference, that's all on the Assay tab. So when you download that, you get that information. So I'm going to click here. So there are two versions. There's a regular CSV file, but there's also a CSV with IRIs for those of you who are interested in having IR information. but I'm gonna just download the basic version. So this will spin here, it'll show you.
What's an IRI?
An IRI is more for AI, so it's for machines to talk to one another. So it gives you actually the web location. Bjorn probably would do a much better job of explaining why.
It's a generalized form of URI, so the kind of web address is a URI, IRI is a generalized form of that.
Okay. I
Yes.
noticed that there's a question mark tab there that explains that the export to CSV file with IRIs, if you click on that, it says it exports results to a CSV file with IRIs.
That is not useful.
Make a note of that.
It's accurate.
All right, so, thank you, but we can always be more explicit in our help buttons. All right, so I'm gonna open this and show you the Excel download. Now, bear with me, it's very busy because it's got all the fields. All right, so the first thing I'm gonna do, let's see, I'm gonna maximize this. All right, to make that big, all right? So, hopefully this is looking a lot nicer with 3DS, it's nice and big up there. It's tiny on the screen. So, each column is a field, all right, and each row is an instance of data, all right? And in this case, I downloaded, I believe, the Tcell tab. Let me look at that real quick. Yeah, thank you. There we go. Yeah, so I downloaded the Tcell tab. So whatever tab is open within the subset of tabs, that'll be the tab. So you'd have to click on Tcell versus Bcell if you want to get those two. And I'll go back and show that to you when but what I want to do is spend a little time familiarizing you with this spreadsheet, because many of us are very familiar with working with spreadsheets. So this has all of the data from that query in this spreadsheet. So if you get familiar with the fields, i.e. the columns, you can do a tremendous amount of work just from the spreadsheet, because that's where kind of the rubber meets the road. The results page is showing you a summary. This is actually all of the data we downloaded, all right? So if you look here, this is organized. The first part, just like we showed you on the detail page, the first part is giving the information about the references. So this is the authors, the PubMed ID, so on and so forth. Then going to the right, you'll see this is information about the epitope. All right, here. And I'll show you more detail there. Then you'll have information about the host here, going this way, so about that, and then if you keep going this way, all the details about the assays and the responses, the effector cells, whether it's class 1, whether it's class 2, the antigen on the plate, the annotation, it's all there. So this document, this file is very powerful because you can do a lot just from it, and let me show you what I mean by that. So if you go here, many of us are familiar with using functions in Excel. So if you go to this, which is the row that represents the fields, and I'm going to say, let me filter on these columns. So now I can go along the top and I can say, okay, well, so this is the field, this is linear peptide field, so this shows you, this is the field that shows you all the epitopes here. This is the proteins that they came from. You can quickly come here and look at all the different proteins. So you can look at just envelopes. So you can unselect the rest and just select envelope if you're just interested in envelope data. Similarly, you can come this way. And again, remember, this was everything. So you can come here and you can say, just show me human data. So you can unselect the rest. Or you can look at just the macaque data or just the mouse data. So you can do all of that from right here. You can also look at it's all scrunched up, but you can look at the immunization processes. There's the disease is there. So you can say, let me look at, so very quickly in a snapshot, I can say, what are all the administration processes that have been documented here? So there's been administration, there's documented exposure, there's actually occurrence of disease. So there are all those immutable processes there. Okay, and similarly, you can go and filter on specific aspects of the assay. You can look at all gamma-interferer data. You can look at all cytotoxicity assay. And this is by measurement. So this is the measurement group. This is the assay method type. So you can look at that. Where the data is located is there. You can download positive and negative data. For example, if you're doing validation, you would want to unselect and get the negative data so that you can sort on positive. So you can sort on negative data if you want to look at that. Another neat thing you can do here is you can, from here, you could say, just give me all class 1 data. And so you can filter on just the class 1 data. And also you can, again, similar to what you can do from the specialized search field, you can also do here, you can say, I want to just look at CD8s or CD4s, and you can do that from here. All right, so that's a proper way. The other thing, let me show you one more thing and then I'll answer questions because I don't want to not show you something that's important. The other thing that's really cool is that the response frequency is here. So the data that is plotted onto the Immunome browser is here. So you can also do that. You can come here and say, I want just the data that has response frequency scores. So you come here, get rid of the blanks, and you have only the data that has response frequency, and you can sort on high scores, low scores. So you get the idea. There's a tremendous amount you can do just by downloading, exporting the data. And by doing it in the data tab, you have all the reference information, so you can go back to that, which is powerful because if you just look at the epitopes by themselves, yes, you can go back and see what they're related to, but here, it's all there, which is a really nice way to do it. And if you're really good in Excel and really good at writing different functions in Excel, there's a tremendous amount you can do analysis-wise on this. Yeah.
Yeah, how do you do that in Python?
I am not I'm not a programmer, but you can. We do if you download the database, you can write scripts. Yeah. So if you download the database, you can write scripts in Python. We do SQL, so we have a SQL version, and I do have experience doing that, but Python I have no experience with. But we have informaticians who do, so you can chat with them tomorrow, and they can give you some ideas.
And I might note that you can take this CSV file and read it directly into R, if you use R, and certainly Python is another option. It's a parsable file. It's
import
data. You can do whatever you
CSV.
want.
Just import CSV,
Yeah,
read
import
it in,
CSV.
and then you can do whatever
But
you want.
you have the multi-level indexing here, so.
Yeah, you skip the first line and lose that.
Yeah.
Or that's at least that's one approach.
Do you have web service APIs to pull this type of data?
We have, yes, we have, you can download the data, just the tools. Just the tools.
APIs, no. For the queries, no. But you can, I think if you really want to do more computational work, the best thing is what Carrie was just saying, just download the whole database, have your own SQL instance and work against that. That's the most.
Again, really good questions, you guys. All right, so wait, let me go back to. All right, so that was showing you how to download all the data from a particular tab. In this case, the assays tab, which I think is where the rubber meets the road for many of the analysis you might want to do. All right, so now we're back on the results summary page. I'm going to get rid of some of this because it's starting to get super busy up here. All right, so now our initial query was just dengue virus. So now we want to refine that. So I'm again showing you how you can use these search interface to, now I'm going to look at just human data. So I went down to the host search interface and I just clicked human. So you can see the human data is about maybe a third of that total. All right, so again, we're looking at that. You can then take this human data. So this is a query for just human data, and you can run an Immunome browser plot of that. And that'll take a few seconds to come up. We should have music. I'm convinced we should have some music with this one. Here it comes. All right, so there you see the live version of the Immunome browser. Okay, so I'm going to just spend a little time walking you through this. So up here, really nicely, you'll see it also copies your input criteria. So it'll show you, it reminds you what you've just mapped. Because sometimes if you do several, you might forget. This tab also will stay up here, so you can go back here and do another query. So this tab will stay up here. All right, a couple of things I want to show you. So if you mouse over, you can hover over a particular residue. It'll tell you what residue, and it'll tell you the upper and lower bounds of the response frequency score. All right. Another thing that's cool is you can download this image. So if you run a query, you want to include this in a manuscript, you can download this. It can be saved, it can be annotated, so there's several features there. Same thing in the lower graph. Another neat thing is if you're interested in just the data for a particular protein, so say I'm interested in the envelope, so you can zoom in there. And then you say update display. So now this data is just showing me the epitope mapping for the envelope protein. So if you can know the range of your protein. Up here, it's reminding you where you are in the big scheme of things. So that's what this gray is showing you, that the envelope is right here. This is down here. All right. So then you can reset that. All right. Then you go back to the beginning. See, if you scroll down here, you can then export the data that's been mapped. Open that up. All right, so again, you'll see all the information here, and you can filter on that. So you can look, so you have the upper and lower bounds here. You have response frequency here. All right, you have the epitope and its position. So you can sort, so now once you're in Excel, you can do all sorts of things. You can sort high to low. You can do the same thing with the response frequency score. You can say, I don't want to look at any negative data, so I only want, or I want it where it's not no negative data. So you get the idea. So there's a power in downloading the data and getting that data on your laptop so that you can analyze it further downstream.
I have one question.
Yeah.
How do you do the mapping? Because you have a sequence, that is, let's say, 15 residues, and then you have a count to the response or whatever. So you assign that response to a particular residue in that epitope or to the payload, to the whole stretch.
So if I understand, so you're saying how do the epitopes get mapped to the reference?
No, I know how it gets mapped, but then the experimental data of the response you have. Let's say that you have, I don't know, a frequent response of 5.
Yeah.
So you assign 5 to every residue
Yes.
on that epitope.
Yeah. That is the granularity we can achieve at this point based on the data, because that peptide was tested and found to be reactive in X number of people. So yes, but it's a good. All right, so that'll stay up there. So now I've looked at just the responses in humans. So now say maybe I'm interested in B-cell responses in humans, so I'll just further refine that. So human is still there. And now I'm going to look at B-cell responses. All right, so we see a goodly number of B-cell responses. And maybe I'm interested, so I'm going to go on the Assays tab, maybe I'm interested in looking at structural data. So because you can sort on the assay type, I'm going to sort, and it's alphabetical. So we're going to look at X-ray crystallography, so that's going to show me where some structures are. And again, you've seen this icon here. I'm going to click on that icon because I'm interested in looking at this particular epitope. So I'm going to go ahead and click on that. Make it as big as possible. And it'll take a few seconds to load. Okay. So there it is. And now you see the structure. So this is looking at this in real time. So you can mouse it big, mouse it small. All right. The different colors show you where the heavy and light chain are there. All right. You can say, show me where the epitope is. So this is now showing where the epitope is. And because the way my eyes work, I like to then go here and you can color this. So I'm going to say color. I'm going to change the color of the atoms. And I'm going to say make them yellow. That just makes it easier for me to see them. So now you have the epitope highlighted in that structure there. Okay, you can also then show me where the receptor residues are. So those pop up. And again, you can color these depending on making them just contrast, so I'll make that green, so now they're contrasting. So you can see, and like I said, you can zoom in and look at that, and you can get it in a nice orientation, zoom out, and then if you right click, hit file, you can then export this into a JPEG. All right, so this is just some of the things that you can do that I wanted to show you. All right, so let's go ahead and get rid of that. All right, so now I'm going to go back to just looking at all data, so I'm going to unclick the filter. So now I'm back to just the human data. All right, so now maybe I'm interested in just class 1 data, so I'm going to go down there and click class 1. I'm going to query for that. So this is human class 1 data. All right, so maybe I'm interested in plotting what's known, what's the aggregate data look like for human class 1 data for dengue. So that's going to come up, so you can look at that. You can see some of the reactivity. This is probably pre-M or envelope here. Again, if you're familiar with your polyprotein, you know where they are. So then, okay, well now I've looked at class 1, maybe I want to look at class 2. So I'm going to rerun a query and look at class 2 data. And you see there's a little bit more class 2 data here. So what does that look like? So now I'm going to run the Immunome browser for that. And notice that the initial tab is still up. Your class 1 tab is still up. So now you can look at your class 2 data. Again, it says here, it says human class 2 data, so it reminds you what this is. And you can compare it to class 1. So those are some of the things you can do using the Immunome browser. Very quickly, you can look at the aggregate data and get an idea for where the prominence of epitopes are falling in humans. All right? So let's go back. So maybe you are interested in specifically, again, all data. Let's go back to the beginning, all data. So instead, maybe you're coming at it from a different point of view. Maybe you're saying, okay, I'm looking at dengue, but I'm interested in looking at the different forms of dengue. So dengue hemorrhagic fever. So now I'm going to run a query just looking at those papers that described epitope discovery in the context of dengue hemorrhagic, say that fast 5 times, hemorrhagic fever. So you can see that. And again, then you can map that to the Immunome browser and look at where they fall. And then you can also then come and refine this search and look differentially. So I'm going to come down here, I'm going to click that off, and I'm going to type in dengue and look at just dengue fever and make a comparison of overall responses that have been reported to date for dengue fever versus dengue hemorrhagic fever. And as you can see, there's a lot more data reported for dengue fever versus hemorrhagic fever, and that's just a matter of what is reported. It's not to suggest that there are more epitopes for one versus the other. Okay, so let's see. I think I've covered all of that, and now on to the next. Any questions with this? While we're here, pretty good. Good. All right. All right. Nima's going to show you an example now. All right. We're going to go back and forth, by the way.
Okay, so there was also some feedback received that indicated there was some work in allergy being done by some of those of you in the room. So not so much an example as maybe just an overview as to how to approach looking for allergy data. So there are, as you've seen, multiple ways to get at data, different types, but one way to look for allergy data is to do so by disease. So from the disease finder, of course, you can simply use the radio button, allergic diseases, which is very broad. In our finder, if you open this, I'd like to just show you that over here in the tree, under the broad disease node, by expanding that, you can see that there are various branches for our basic types of diseases, more or less by category of data that we encounter and curate. And so there's a branch devoted explicitly to allergy. And the way that this is organized is that it's got sub-branches, and you can see there are like anatomical locations. So in terms of where the allergy manifests, allergy of the respiratory tract, connective tissue, etc., and uncovering those shows you, the specific type of allergy, so you could get to data in that way. There are also categorizations done by what's triggering the allergic response, so we call that the allergy by trigger. And so here you have various categories as well. It could be, for example, drug allergy. If you select penicillin, we would expect to get data in which there was allergic responses to penicillin. And so you can see here broadly different types of specific penicillins, and of course you've seen all the assay information before. So a maybe more direct way to get at it, especially for those of you who study allergens, is to search by the actual molecular entity, the allergen of interest, and you can do so by using the Antigen Finder. And one of you, I believe, was nice enough to provide some examples of specific allergens that you work with, and so I thought I would use that as a demo to show what we have in the database. So this is for parvalbumin. Anybody except for that user know what it is? Oh, I kind of give it away. I better hide this. Parvalbumin? Fish, good. I think good job. That was a learning experience for me. But yes, this is apparently quite commonly occurring protein across multiple species of fish. So parvalbumin, here you can see, for example, 3 entries come up, and using the traditional allergen kind of nomenclature, we have 2 specific different species. I think they're both types of Atlantic salmon, but I'll just use the GAD-M1 as a go-to. So now we're essentially saying, show me all epitopes that have been reported from this allergen. And so clicking that, of course, we would expect that our allergen tab would have one result, but here we have the 50 some odd epitopes. Again, because we chose a very specific antigen, they're all from that antigen, from that organism that's the source of that antigen. And here they're characterized in multiple references. So going into, I think it might be interesting to also show you how I showed you before, getting down to the assay details, to again, just get some more familiarity with how to maybe interpret what's in these fields into kind of a narrative of what the paper showed. So in this particular example, we had human with documented occurrence of allergy, so that in vivo process type was captured as occurrence of allergy. And the immunogen source organism is captured here. You can see more information about the patients. So this was an antigen inhibition assay. But one thing I wanted to point out here is that if you go back, and if we take the immunization field as Carrie demonstrated and sorted, you can see that everything we see on this particular view is showing us allergy. But if we kind of probe for what else might be in there, you can see that there are some administrations, there are no immunizations whatsoever. And so the question is, what's going on? Why are we expecting to see, or why is it not unusual to see more than just hosts whose in vivo process was occurrence of allergy? So one answer to that question is that in the context of allergy specifically, we capture both allergic patients and the so-called normals when they're reported, as they very often are in that literature, side by side. So the same exact assay, the same exact epitope might be screened in normal patients versus allergics, and then the responses will be reported for those groups of patients. And we'll capture those side by side. Again, we try to, as much as possible, not be subjective about making the call of what's positive and what's not positive. So by having, very often in the normals, there will be response frequency that's not zero. So by having these two side by side, it allows the user to see, you know, kind of full disclosure what the paper said and make your own sort of assessment of maybe what you might consider positive or not positive. But if you don't want to see the normals, for example, you can easily also filter those out, you know, again, by exporting this data and either sorting or doing some sort of offline computation, but it's quite easy to actually make your query specific enough to avoid that as well. So you would simply just, for example, if I did the exact same query, so these are B-cell epitopes, if I did the exact same query with a detailed search and specified that same protein, you could simply just say, I want part of the albumin epitopes from GAD-M1, but this time I only want patients who were allergic or donors who are allergic. So the in vivo process type, you scroll down, occurrence of allergy, for example, is there, and I think we had 51 epitopes before, something of that nature. I don't remember exactly how many epitopes we had. So nevertheless, if you go here now to the assay tab, everything should be in vivo process type of allergy. So we've sort of successfully sifted out that data that's either not relevant to allergic donors or was in some way done in, you know, some other sort of model. So that takes care of that one. Any follow-ups, questions? And you know, with the allergy data, so we encourage you that are interested to come and see us and talk to us about it. And the breakouts are one-on-one. We can show you, you know, kind of more strategies and how to explore it. But this is just an example. Yeah, that's right. Leora, who you were introduced to earlier, is the kind of go-to curator. She has the most kind of hands-on expertise with this data, so she has a very good intuitive sense as to what's in the database, and very good at also when you get the results of interpreting it. So she's a good go-to for your allergy studiers. All right, I'm going to turn it back to Carrie.
All right. Yes. All right, so I'm going to switch gears. I was going to say this for later, but I want to make sure I get to it. So I'm going to show you a little bit about TCR-MHC interactions. So there were some questions about that, kinetic measurements, and looking at the data used in that way. So I'm going to do that. All right. So we're going to look at, we want TCR-MHC interactions. So I'm going to open up the Finder for assay. So this is a thing you get at through the assay. I'm going to open the Tcell assay because this involves TCR, and I'm going to look at the 3-day structure paper. This is as a first glance, all right? So I'm going to apply that. So I'm opened up a Finder and I'm not using it, so I'm going to apply that. So now that's applied here. So now I'm going to hit search. All right, so you can see this is epitope data related to TCR, MHC. All right, so we'll look at that. So if you look at the Assays tab, you'll see that there's X-ray crystallography there. So I'm gonna, again, open this up. So you can look at any one of these. I'm just opening this first one up as an example. So we're gonna look at this structure. So I'm gonna open that up. So it'll take a little bit of time. You remember with the B-cell structure. So this will open up. You'll see here. All right, so again, you can familiarize yourself with this. You've got the MHC, alpha and beta 2 microglobulin, excuse me, it's a mouthful. Then there's the TCR alpha beta is here as well. You can click to highlight where the epitope is. Again, you can color that same way. I like to make it yellow. It's my issue. I need I need it to be bright so I can see where it is, all right? So there's that. So all that information is here. This is all calculated from input from the sequence that's in the curation, all right? So that's a way you can very quickly look at structure data, all right? So say you've looked at that structure and that's one of interest, and you might think, well, I'm very interested in looking at what kinetic studies have been run. And a really neat way of doing this, and we haven't shown this to you yet, but there's been a tremendous amount of work setting up epitope summary. And the way you would get at it is here. So you can click on this epitope, and very quickly, this summarizes that particular epitope. So you can see what alleles it's been associated with, and you can click on this and go straight to that binding data. But also you can do this. You can go to dissociation constant, so you can get at kinetic data for that particular thing. So this shows you very quickly how you can do that. All right. So then you can click on that, and that links you to. It's very important to point out that because of the, forgive the term, the granularity of the database, you can get at things from multiple different ways. There's not just one path to get at some of the details. And so it takes a while to get used to that, but we have ways to do that. So now you've got this epitope and all the assays associated with this epitope, including ones that define kinetics. And you can, again, you can sort on that and look at that. And you can sort by MHC. Again, you can download this and look up that. Let's see, we'll sort on post. All right, and again, because it's TCR that. So that's one way you can get at TCR, MHC interactions, look at them from a structural standpoint and look at them from a reactivity standpoint. All right. All right, so another search I wanted to do, we don't want to be too infectious disease heavy, so I'm going to do an example of autoimmune, so I'm going to do type 1 diabetes. So I'll touch on a few things. One of them will be how to look at patients specifically, so again, this will be kind of a clinical status search. We'll look at how to look at autoantigens, how to look for autoantigens, and how to compare different, so a mouse model of diabetes kind of thing, all right? All right, so again, from the home page, you can come down here, and again, I'm interested in diabetes. So I'm going to start typing. You can see the autocomplete gives you these choices that match that hit. So there's pre-diabetes syndrome, insulin-dependent diabetes, and there is some type 2 diabetes data in there. There are very few, but there are a few instances of that. So what you can do is you can, I would suggest, again, if you look at the high, looking first broadly, I would include pre-diabetes. And so this is additive again, so you can stack these. So you can add both. So I'm going to do insulin-dependent and pre-diabetes. And I'm going to simply do a search there. So you can see that data there. All right. And because this is an autoimmune disease, you might be very interested in perusing what self-antigens are here. So again, you have the antigens that are associated with pre-diabetes and insulin-dependent diabetes, so type 1 diabetes. And this is a summary of the relative abundance of each. So there's 192 epitopes from GAD2, maybe not surprisingly. You can then look at that data using the Immunome browser. All right, so you can see the different reactivities over the length of that of GAD2. That's human GAD2. Again, you can go down here and look at specific. I didn't spend a lot of time with response frequencies, but again, the closer to 1 it is, the better it is. For example, this here, this is 0.5, 0.45. And again, if you download this in 6.2, and what you'll find often, if you're familiar with this area of study, you'll know that the well-studied or the well-characterized epitopes will fall within these regions that you see in higher activity. All right, so we've looked at that. What you can then do, you can look at that. So there's proteins here that you're familiar with if you're familiar with type 1 diabetes. These are self-antigens here. So then you can go down and you can say, well, I'm just interested in human data. So you can click on human data. And again, this is just repeating some of the similar ones we did before. All right, so again, we've got that. So it would be interesting to look at the mapping of, let me get rid of this, that. So we're going to map the human GAD2 data to the email browser. All right, so this is the characteristic. So this is the aggregate data for human data, human responses to GAD2. And this is both D and T. All right, so now I'm going to say, well, let's look at the mouse data for that and see how it compares. So I'm now going to go down here and repeat the query, but selecting mouse, and then we'll go down to the same antigen, so GAD2. And you can see a very different profile here. So this is the animal model. This is the human data. So you can very quickly look at the level of aggregate, the differences between those. So that's one feature I wanted to point out to you. All right, so say you're just interested in looking at looking by antigen. So say you start on the home page and you're interested in just insulin. I will tell you, let me back up here, that when you're using proteins, you're looking for antigens or data that is very rich, that is, there's a lot there. Here, being specific can help. So if you say human, so you're saying I want human insulin, especially with, so you look at the high node for human. We didn't mention already, the high node, that is the, you probably saw it when we did hemagglutinin as the example, there's 2 stars next to it. Those are the high nodes, so those would be the ones you want to select if you want to look at the entire parent node. All right, so I've hit looking just for insulin here. So I'm going to run that query. All right, and so this is insulin from humans, and then I'm going to say I just want to look at human responses to that. Good number of epitopes. It's actually a very small protein for those of you not familiar. And those are the responses that have been, so these are some of the heavy hitting immunodominant epitopes, 80 to 93, so you can go down here. So around here you can see here and here sequences containing these cores with really high response frequency scores. In this particular one, this was tested in a very large number of people. So it's nice to see this raw data because you can get an idea for whether the N was 5 or whether the N was 100, because it matters in the end. So I think that's done. Are there any questions of this particular query process? All right, I guess Nima, we can
go forward. I did, sure. I mean, the rest of them were those post-lunch ones. That you want to go forward with.
Yeah, we can do that. We can also do, oh, I know. One thing I wanted to show you, I'll do it now. So as we alluded when we were first, all right. So at the very outset, I was trying to convince informaticians that they can make use of the database. And I was saying that what you can do is you can validate your data. And for those of you who are not Excel wizards, or maybe you are Excel wizards, but you haven't necessarily applied your math skills to looking at analyzing data, you can download a spreadsheet of positive and negative values from, say, a query on T-cell responses in dengue or whatever virus you're interested in. And then you've subsequent to that, you've done a prediction, and you've got prediction results for, class 1 CD8/class 1 epitopes for a particular protein from that virus. You can then have one tab being your download of validated experimental data from the database, and the other tab being a list of your epitopes that you predicted, right? So then what you can do is you can do a match function. So you can add in here a match function, which basically says, tell me every instance where epitopes from this column match epitopes from the data. And it'll tell you which row. And you can do it the other way, so it can tell you which row. The power in that is when you have a match, you can see if that match was positive and who it was positive in. Was it positive in humans? So that's a very quick way, that's a very simple thing for an informatician to do to validate your prediction results. And so instead of synthesizing a bunch of peptides that have already been shown to be negative in humans, you could start with the subset that have already been shown to be positive in humans, or ones that are related to the ones that have been positive in humans. Does that make sense to everyone? So that's a very quick thing you can do in Excel by running a match function. And for those of you who don't know that, I've got it written down, and I can send that to you or show it to you during lunch or during lunch breaks. All right. So, all right, we've still got some extra time. Does anybody have any questions? Have an online question? No? Okay. All right. So you have any examples?
All right.
All right.
Yeah, well, I can do.
Okay, for good measure, just because I've taken you through a B-cell specialized search, a brief T-cell specialized search, and then that way you can see some of the quote-unquote buried fields that are accessible but not necessarily found on the homepage search. So let's suppose I want to formulate a query where, as I showed on the B-cell side, I might be interested in only monoclonal antibodies. So sort of the analogy of that on the T-cell side might be a specific phenotype of an effector cell. So you can define that easily through fields on the assay cluster fields as well. So let's say we can pick another organism that I think stands out to me as having come up on the surveys that you were sent was, there were a couple of you that talked about malaria. So we can just do regular in the context of Plasmodium falciparum. Of course, if you don't spell it right, you won't get it. Okay. So, defining whatever you'd like in this, I'll just, for sake of consistency, we can use humans. And then, again, you can specify if you're interested. Like Carrie said, one way to approach this might be not to do over query to start unless you have a very specific query parameter or set of parameters in mind. You can sort of start broad, explore the results, and then delimit as you'd like. So we could just keep it broadly based on any human. But what I wanted to show you is that down here, one of the things that is sort of, again, a standout distinction, a very powerful aspect of the IEDB, and the fact that we have manual curation affords us the ability to very specifically say what cells were tested in this T-cell response. And so that's there for you to kind of exploit, either on the effector side or even on the antigen presenting side. You could define what cells you might be interested in. So if we expand this effector cell cluster, you can see that the parameters of the effector cell that you could define are either tissue, the actual cell type, and then interestingly, what we also offer is how were these cells, if at all, re-stimulated, or how were they treated prior to assay? So are you dealing in what's the clonality of the cells? Are these polyclonal? Were these tested directly ex vivo, let's say with an immunization, and then the cells were taken out and directly assayed, or was there like a period of re-stimulation in vitro in between? Or was there a cloning of the cells so that you're dealing with a specific T-cell clone? And so all of those things can be of interest, and they can be specified in the culture conditions, much like on the antibody side, so that you're excluding cells that may or may not be polyclonal, for example. So But in this case, I just wanted to point out that basically the types of cells are very broad, and you can see again there are multiple checklist style where you can select one or many. And in this case, I'll just choose a T-cell CD8 positive, and we can go ahead and see that now confirming that everything that comes up on the assay side will be a CD8 positive response. And so you can see that in the assay details. If you go down to the effector cells, here, these were in this case T cells from the blood that were assayed directly ex vivo. And so, you know, just playing around with it, you can, like for example, one question that comes up a lot is interest in tetramers. If you have a question as to whether a tetramer has been utilized in the context of all these positive epitopes, you could simply go into the assay and say, now I want to know all these positive CD8 T cell responses for which there's been a successful use of a tetramer. And so the assay tetramer is in our list. This would be here, and then by deleting that. And I chose positive only, so any results we get should be actual cases where tetramer outcomes were positive.
I know.
I know there are some. Anyways, we're going to let that roll. So then, as you explore here, I just want to point out some of the other fields on the antigen side, because again, I had not done that. Again, here, the MHC allele is available. So in this case, if there is a restriction defined for the response, you could, and you're interested in certain restricted responses exclusively, you could specify that here using the finder, or just a broad category. I don't know what's going on with that query, so we'll just let that run. There it was. That was, as I mentioned, just to keep the balance, showing you the power of the T cell side as well. Please, any questions?
So, do you have an expansion of other markers that are on the, say you want a particular marker combination like CD8 positive T cells with PD-1, is that type of functionality available?
We don't have those particular ones available. So one of the things that we often find is that the focus of the paper might be to characterize the phenotype. And in those cases, if they're single phenotypes, we do capture them. We have some combinations, right? Let me bring that list back up. So, what's that? Yeah, they're just like CD4 minus CD8 plus, but
Okay.
not two different phenotypes as you indicated. We do
Okay.
not have those. But if they are, the response is attributed to a single one of these markers, we will capture it. There's anything else? We have a little bit of time to go, so we can do, what's that? Yeah, it's still going. Somehow I caught it into it. Yeah. You want to do another one? We still have 10 minutes.
Yeah.
All right.
Well, we can certainly do. Review that it's not broken. Doing a tetramer query. All right, so tetramer query. This is all tetramer data in the database, because I just did tetramer. So again, you could sort here, which will, so you can sort there. Look at tetramers this way. This way. So a lot of of tetramers, not surprisingly for influenza A, targeting hemagglutinin. And then, of course, you can look at that, and you can look by allele if you download this. Obviously, the easier way to do it is specify the organism, but just to prove to you that it's not broken, I wanted to do that. All right, for those of you who are interested, we don't spend a whole lot of time on this, not because we don't care, but because it's such a small portion of the body of data that is epitope data, and that's transplant or allo reactivity. So I just want to show you how you can get at that. So if you open the disease finder, one way you can do it is click on the disease, and this will allow you to peruse all the data. So there, that shows you that. So I selected that, all right? So there it is up there, and I'm going to hit apply. And then I'm back on the search page. I'm going to hit go. All right. So this is all of the epitopes that have been defined in the context of transplant. And so you might be interested in looking what the alloantigens are. Again, I'll sort here by epitope number, because the default is to sort by the number of references. All right, so some heavy hitters, not surprisingly, would be for hemophilia. All right, others are class 1 and class 2, and so you can look at that data. You can map those, obviously, we showed you that. Let's go here. I'm going to see, let's look at just the data for factor VIII. So now that should be set for all the data that's just related to factor VIII. So again, you might come here and you're interested in transplant because you're interested in alloreactivity in general. You may not have a specific one in mind, and this would be a good way to familiarize yourself with what's been published so far. We're being attacked, we're not. I know this is going to sound ridiculous, but this normally doesn't happen. All right, well, fine. I wonder if there's somebody we should enlighten us. This is always the thing you worry about when you're doing live demonstrations for a workshop, is that the database gets hung up. All right, does anybody have any specific questions while we're waiting for this to?
Yes, for the transplantation, are you looking at epitopes for allograft rejection?
Yeah. Yeah, so, actually, you know what I'll do? Let me do this.
Let's see if they're
Yeah. Yep. Well, I'm not.
Great. Yeah, exactly. It's a good time for lunch.
Here we go. Okay, so yeah, we can take lunch. We're good, but essentially, what you can do is, you run that search and you see the different autoantigens that come up. Again, you sort by abundance here, and you can look at that. And so, say you filter on one. Oh, I did it again. All right. Oh, work this time. Yay! All right. So then, this is human data. So what you can do is you can look here and very quickly see that this was a transplant recipient. Serum for patients with hemophilia, treated with purified convent factor 8. Right. So that would be how you would get at a particular aspect of the response to the human epitope in the context. Okay, so I guess we can go ahead and have lunch now that we've exhausted you with spinning wheels. All right, if you have any questions during lunch, feel free to come up and talk to us. Questions. We are going to
We are going to the back, the far side of the back patio. So we'll kind of walk out as a group so you can all see the room. You're welcome to leave your stuff in the room. The bathrooms are still on the other side of the building. Yeah, you may need to kind of use badge access to get in and out of the building. So feel free to grab one of us or bother any of the people sitting outside for lunch or whatever. But yeah, there's a spread set up so it'll serve yourself. So when you get out there, you can just make yourself plate. Yes, let me look at it. Yeah, I'll just bring it back. Yeah.
With regards to epitope condition.
Yeah, it's so big, right?
India way. Well, or the bricks.
That's what I mean.
Oh, cross. There's a guy that come out the other way. Well, I can say that. So, I mean, any more now. It's wrong.
So what's there's a difference. Yes. So how we stay
and then there's two.
So we
Did I say that?
A couple changes. Swap these two slides.
Oh, yeah. So I just need to get myself copied. Other things, or the thing was, and it's not so critical, but, you know, we're gonna say, it's on slides 9 and 20, the URL is not what is that? Zero flag.
So I once saw,
There's a reason why I'm telling you. Yes, I love it. I just bought a bunch of from a festival so that my 13-year-old can start on that.
yeah, we saw it's really a one-man show. It's great. See, it is paired differently and then together.
So let's
Very-
say, here we are for the next paper.
Okay. Okay.
It's fantastic. At one point, it's just one. And it's very- So I have discovered that your previous ballot had polished the black shoes, so brown.
So what they do is they publish a huge supplement in Excel of all the peptides. And they'll say.
Is that all you're gonna say?
Can anything be done? No, sir. I have given them to the gardener.
Oh, they've been given, okay.
Anyway, so I'm sorry, no, but.
All right, that's good to know that they're not being.
Do you guys capture their attention?
No, I don't know.
They're being still, okay. Okay, but you've given them, that they're on the.
Yeah, but we can, no,
It's not important. I know that it's our kind
Just because, I mean, and it's funny because
Well, it's, the reason is because kind of us to say, I started.
Yeah, okay.
You know, if you designate.
Yeah. Which is only step one, right?
and look, there's a link, yeah, bang. we can
But
go there.
then there's two of them that.
Yeah.
I would just say, link Siri, sorry, it's not taking.
Yeah, I mean, yeah.
Yeah, that would be.
That's
yes,
that would be.
that would be.
And then I would go ahead.
So you guys don't,
Do it, but I already sent the slides to Sharon this morning.
No, that's not, no, it's not worth that at all. So, good, as long as it's not like false data, which this kind of is, but that's okay.
As long as that, what?
As long as that's not false data.
Yeah.
Which this kind of is, but it's
Well,
not critical.
No, it's not false data, it's just
Not
it's
critical to the science of
it's
it.
an error in the presentation.
That's correct.
But it's still, but the link, I mean, if the link were broken,
It's so complex, it's
that would be
definitely not my
false
area of
data,
expertise.
if the link didn't work.
But
But still,
Still cool.
yeah,
It's
well,
just
it's
showing
not
us up
code.
to be
It's not code.
inaccurate.
Well, I was told that, well, put it this way, an Iranian rug
no,
merchant.
but once we link with them.
Excuse me?
An Iranian rug merchant.
I heard, you know what I heard? Drug merchant.
Oh, rug.
Because there are those too, you
I
know.
know, one's told,
Hopefully with next year, I mean, that's.
every rug.
next year it's coming.
I know, I swear, the year is going by. Right, so,
Ah, this one.
so.
Okay.
289.
289.
I don't know why it's been spinning the whole time I've been in here, but again,
Yeah,
like
so
I said,
that's,
this is
and
a new computer, so
he's.
I think I can do
Okay.
it. I can do that, it's just such
Really excited because he really wants to coordinate.
Yeah.
This community, so the community does exist. Okay.
But it looks like if you're looking at the logs,
Yeah.
It's
showing an
He reached out to us.
Oh, I
To
see.
get us involved, and we said, yeah, of course, we want to be, so you can get that, that's just
Yeah.
fairly recent, I think it was just last year.
I just want to see
Yeah,
what
really
kind of
excited about that.
information I can
There's,
get.
he's super,
Yeah,
super.
and then
Yeah, I heard
the
these people
tools, which all the bioinformatics guys.
workshop, and I think he was
Quite powerful.
Yeah.
Quite.
There's a slide
Research
show
a lot of stuff.
we don't know what I saw.
It's very unique.
Yeah, I mean, we're definitely working
Yeah.
on it, I mean, whatever, you know,
Okay.
we spend a tremendous amount of time just reaching out and making sure we are linking to other resources, because, there's a lot out there. And that just makes it more useful to people, because they can come and link out, link back in, you know.
Yeah. Another question about is
everybody we got for lunch?
Yes.
Although there's no vegetarian sandwiches. You know, people walking up and saying, oh, do you have any vegetarian sandwiches? No, just turkey. So that was the only flaw, so that's not bad.
Okay.
Now these people starved to death, but there's veggies, and there's fruit,
Okay.
and so
Okay.
there's salad, so there's things that they can eat. So if they're gonna go and get lunch tomorrow,
Yeah.
then it needs to be
yeah.
specified,
I need
because there's a lot of
to
Indian individuals in there.
Check with, yeah. Okay. I will.
And then, do you want me to call Einstein and change the, coffee
I
for
just
tomorrow?
texted Nicole.
Ah,
I don't know who has the confirmation stuff or whatever.
well, I have a copy of the,
Okay.
what's supposed to come.
Yeah. So then, yeah, if you wanna do it, that would be great. I will bother her about tomorrow's lunch and vegetarian options. And then reception, starts at.
5.
Reception starts at 5.
And there's no faculty meeting, so they should all, Bjorn and Alex should be here.
Okay, cool. Yeah, so
Cancel
let's,
that.
so technically our session ends at 4:40. So I assume people will kind of linger, do questions, you know, take a while to get out there, but they may also go early, right? Like who knows how the end of the day is gonna work. So maybe we should try to have everything out there starting around like, or by like 4:20 kind of a thing.
I would
That
say
way.
4,
Yeah,
because they did not get any ice.
Okay.
And so we found old ice
Okay.
in the refrigerators down here,
yeah,
but it was too late. By the time we brought out the ice, they were already starting to serve themselves lunch.
yeah, yeah.
So I just took one of those things and filled up the ice with a scooper,
Okay.
so they can at least scoop some of the
Well, and
ice
I'm
in
also
there.
gonna have,
So-
maybe Jose run and get ice.
Yeah, he's aware of that, because
Okay.
you filled up your cooler.
Cool.
The remainder of the beer, 1,
Yeah,
and, all of the red wine is
Mhm.
underneath the table.
okay,
The cooler is full except for with ice.
okay,
And so now just sodas and water
okay,
needs to be found.
yeah,
And Jose said that Nicole went over and got his Pepsis and stuff like that. So
Okay.
I'm guessing
yeah.
that she's gonna get ice, or have Jose pick up a couple extra.
Okay, but I will make sure with them that one of them goes to the store and buys ice and brings it back and then fills.
And starts cooling it now.
Yeah, yeah,
Not this wait until the last minute
yeah, yeah.
deal.
Okay. Speaking of,
Yes.
haha.
Run!
Okay.
I know.
Yeah, right? Perfect.
So you may have to pick up more than what I suggested, you know, the 1.5,
Mhm.
you know, because that was what we filled up,
Yeah,
but I think we need more.
okay.
Yeah, so,
Because-
and I'm thinking what we can just do is put all of the beer and everything in those buckets that are on top of the refrigerators and just throw ice on top of that to start cooling that now,
Okay.
right?
Well, we did really good with the beer and the wine, so we filled up all the cooler.
Oh,
And
okay.
then one of, like,
Yeah.
my size tub.
Yeah.
That's underneath the
Yeah.
seat.
Yeah, oh, and that's everything?
No.
No.
Oh, okay.
And then there's red wine and a box, one box
Yeah,
of beer.
yeah.
So that's
Yeah.
it. You said don't open it unless we need
Yeah, I
it.
was thinking it, but then it's like also if we need to start cooling it, maybe we should just open it and then,
I threw the top we filled into our mini fridge,
Uh-huh.
so it's in there.
Oh, okay.
Just FYI. So
Okay.
you don't think anyone's stolen or anything?
No.
So,
but yeah.
Drink it on your
Yeah.
lunch break. I got it.
Yeah.
he is, yeah.
This isn't going.
But yeah, so let's get everything, anything that's not cooled already, let's go buy however much ice we need. Ice is cheap and easy.
Yeah.
Put it on it, start cooling all of it, and get extra, that way we can put more on it
Okay.
later, kind of a thing, right?
Is there anywhere we can store that actually?
In the freezers down here
Okay.
is totally fine, or whatever.
Yeah, because
freezer
9 times out of
space.
10 they won't take ice.
Okay.
Yeah, yeah, whatever freezer space.
Oh, I
you
have a
can
big
find.
bowl of fruit. Did you want me to
We
save
can,
that
we
for
can
this
put it out
afternoon?
there right now. Let's just put it out with the lunch stuff.
Okay.
Because the vegetarians can eat more stuff.
Okay.
So,
I just piled the rest of one into the other.
Yeah.
It doesn't look too bad.
that's fine.
And, okay,
I trust
so I'll go get it.
Yeah, so, okay, so you put fruit out there. You're my ice man.
Yeah.
Okay.
Ha.
There's layers to that.
And
We
then,
should,
yeah,
so
so
we'll
many
take these
layers.
out of here?
Yeah, well, I was gonna ask Muscles over here to put them out on the table out there.
I'm
all the lunch
actually-
people. Oh, yeah, I forgot, right, of course. And then I
Wait,
will,
put it on the table here?
I was thinking like where they're eating lunch, that way if any of them want waters or anything.
They can come in here and get it.
Wait, you wanna hear that? Okay,
Because
okay.
there's plenty of volleyball stuff out there, and it's just not.
Yeah,
enough
that's
ice
true.
right now.
Okay, yeah, then just leave this here.
Okay.
Okay, so fruit out there, Iceman.
Yeah, I'll let you know one right now, but.
Perfect.
Oh.
I will bother Nicole about vegetarian options for tomorrow, and then you will call Fines and see if we can get the decaf switched to regular.
Okay, you need to stop for just a minute, drink something and eat something.
Hey, I ate that bagel, that was good.
Okay, well then intake some awa, and some, not more
Save all
coffee.
my space for the alcoholic. No.
There's always space for that, even if you just
True.
fill yourself up.
Very true.
I left
Alright.
the keys in my drawer.
Oh, right, yeah, I guess I'll need my keys at some point. Cool. There wasn't, my car was still there was no
There
tickets
was-
or anything?
no, there was-
Perfect, okay. Okay, cool, you're wonderful. I think that's about all we, ooh, the other thing we should see